Hello and welcome to Getting to Know Your Soil with MX Mica Francis, me. My pronouns are they, them, their, or he, him, his. And I have a master's in soil and water sciences. And I'm the project manager at Grosvenor Horticultural, Sangre Chiquito, Trinidad and Tobago. So to start off, here's a picture of a couple of the tropical flowers that we grow at Grosvenor. Alpina purpurata, or Ginger lilies is a more common name. And we have red, white, and pink varieties. And kind of interspersed with our more scientific or informative slides, we'll have some fun, beautiful pictures to kind of break up things and ease your mind as we flow through the presentation. So to start us off, I'm going to locate us a little bit. In Trinidad and Tobago is the farm, and this is where we're going to be talking about in particular in general. And, just a little note for like local tourism or even tourism from abroad to Trinidad and Tobago. A lot of folks in Trinidad like to go to Miami. It's about a 4-hour flight, but, the opposite is true. Folks from abroad can come here, or, we can participate in local tourism. You can come and do a tour at Grosvenor or lots of other great ecotourism sites on the island. So now a close-up of Trinidad and Tobago, and then more specifically Sangre Grande, Sangre Chiquito area. Grosvenor is just a little bit south of Sangre Grande proper. Here's another flower. Here we have the Rostrata. It's a member of the Heliconia family, and I hope you can see my mouse here. This is the actual flower, those little bits there where my mouse is, inside of the floral bract, which is this red with a yellow tip, specially evolved leaf, which actually lasts a long time with the flower inside of it. So these tropical flowers, a lot of them are developed to last on the flower for a really long time, or to last on the plant for a really long time. Okay, so now let's think about soil. So the soil here, we have a legend of sampled soils and different soil types in Trinidad and Tobago. So the white area is areas that have not been mapped. And so for the Grosvenor area, we're going to say it's around here. The eastern side of the island. And it's this brasso heavy clay. So we'll get into what how you define a brasso heavy clay and how you make a soil series mapping altogether. But here's another beautiful picture of some flowers and a hibiscus, pink and red hibiscus. I call it Blossom. This one is right opposite to the door of the main farm building, or one of the farm buildings at Grosvenor. So now here, let's look at how we define a soil profile and how we get a soil series, right? So the soil series is really based on sampling the top 2 meters of the soil, and then you see a variety of soil horizons in doing so, and the characteristics of those horizons are what we use to distinguish one soil from another. So as you can see here, the O layer is the top layer of the soil. It's organic matter, and some might even say it's not quite true soil, but it's the organic decaying stuff on the surface. And of course, the O horizon and all the horizons that I'm giving you here, these are the main soil horizons. The A is the next one down. It's generally a zone of loss. And so, sorry, all of these horizons can have sub-modifiers to describe them more thoroughly, but for our purposes, this is more than sufficient. Pardon me. So then, yeah, you can have the E illuviation layer where even more leaching occurs. It's often lighter in color, as you can see here on the figure. The A would have inputs of organic matter from above directly coming into it. And then the E is really where leaching is happening. Leaching is the downward movement of nutrients and carbon and clay in solution. Well, clays would be another specific term, but leaching is generally stuff being dissolved and then moving downward to the soil profile. B then is the subsoil layer. It's generally a zone of accumulation where all of those same silt particles, clay particles, dissolved nutrients then begin to accumulate. And according to the conditions of the soil, how much clay you have, all this jazz, how much waterlogging occurs, you get all kind of different stuff happening in your B soil layer. And you can have your unconsolidated parent material, so your loosely broken up, weathered parent material, and then solid rock underneath. And so the parent material or rock can be all kinds of different stuff. And we'll talk about the potential impacts that can have later on a little bit. So here's another beautiful tropical flower. This one is a golden torch. Okay, so we've talked a little bit now about soil classifications, soil horizons, but like more broadly, almost to take a step back, what is the soil? And the soil is made up of 4 parts that we're going to talk about here now. So the first of these is the mineral soil, the solid particles that make it up. And so we consider the fine earth fraction to be anything under 2 millimeters in diameter. And then from there, you can get smaller and smaller particles, as you can see broken down on the side there. And then you can even get further into nanoparticles. So then, of course, soil also has water in it. There's soil moisture with dissolved nutrients often. There's a lot of air in the soil. When you think about the soil as an ecosystem dynamically, it is really full of lots of pockets that are either waterlogged or not. And if there's not water in that pocket in that space, it's usually air. And then organic matter. So you've got waste, residue, metabolites, live organic matter, decaying organic matter, which is high in highly reactive generally. Lots of stuff, either chunks breaking off, ions interacting with the soil, organisms exchanging one thing for another, usually trying to get energy and so metabolism is either catabolysis or anabolysis, breakdown or combination of smaller molecular bodies. So here's a kind of a graphic. There was a graphic I really liked, and so I've recreated it here to show you like a close-up of the soil, right? So here you can see the macropores and the micropores. Micropores being much smaller spaces, macropores being much larger spaces, with water in blue moving among them. And then you have all of these mineral particles with the rough edges and different colors that could be different elements and stuff like that. And then organic matter, again, with maybe some of these gray, these darker greens being edges that are decaying and more metabolically active. And with the kind of spongy, irregular edges. And then just a little close up over here, you can think about the forces adhering all of this stuff together. So you can have water molecules with adhering to organic matter or to soil particles, or, you know, each of the organic matter and mineral particles adhering to each other via various forces. Again, we don't want to get too deep into it, but it's lots of cool science thinking about this soil ecosystem at various sort of heights, right? Really close in or as a whole overall. Here's another beautiful anthurium here to, you know, take a breath. Don't get too stressed. Soil science is fun. Lots of nutrients and everything that we need cycles through the soil and then comes back to us in plants or animals. So, so how does soil form? Now we have an idea of what a soil horizon can look like, what soil is made up of, and so like how are things going from solid parent material to soil as we know it? So you have climate as a major factor, both temperature and rainfall. Here you can see a map of Trinidad showing heavy rainfall on the east and less rainfall on the west. That would be, of course, due to our northeastern trade winds coming in like that, and then depositing a lot of their rain and then being less water laden as they move towards the western side of the island. Then you can think about topography, the green in the low-lying areas there, and then the yellow would be the higher land or land wrinkles, mountains, topography. And then parent material, as I mentioned, is really important. You can have volcanic rock, you can have limestone, you can have sedimentary rocks. All of that impacts what minerals and what ions would be in different concentrations in your soil. It'll also, of course, impact your drainage, how water moves through the soil, whether it stays and settles on the surface or percolates down. And then time. So time for all of the above to interact, time for weathering to occur, etc. And then, of course, organisms, both two-legged organisms, humans, and or other organisms in the soil, lots of things like insects or even larger animals like rabbits and tortoises, which burrow, can impact the soil system. Earthworms are another big one that people think of. So here, to kind of put it all together, you can think of, thousands and thousands of years ago, you had just solid rock, and then slowly over time, physical and chemical weathering occurred to get the beginning of a soil layer. As organisms, plants became involved, you can see it rapidly starts to evolve more and more of a complex system. You've got organic acids leaching down, death and decay of organic matter, releasing all kinds of acids that then break up the parent material and or the soil on top of it, and then increase your fractionation, your horizon development, until eventually you have something like what we saw before, which is like a soil profile. So let's go to organisms and organic matter. As I mentioned, they're really a big deal. They're a bit of a showstopper in terms of how soil formation goes and increasing the rate of soil formation. So you have live roots, organisms, and earthworms and insects. You also have dead and decaying organic matter releasing all kinds of different acids and metabolites. And then you have microbes, who are these, smaller than the eye can see organisms that are impacting the soil. And they're often engaged in sort of go-betweens, right? They speed up decay and are intermediaries in electron transfer to release energy. So microbes are often single cell organisms. They have no differentiation of sex or gender among them. They're often just that, budding by mitosis. And one of the really cool things about them is that they can pick up DNA fragments and literally sort of evolve by interacting with their environment. So here we can see just a little scale to show you nanometers, the size of DNA versus viruses, bacteria, simple organisms versus eukaryotes. So prokaryotes versus eukaryotes with true cells, and there's some differentiation that we'll look at there in a minute. So to talk a little bit more about the DNA and that interesting point I rose, I promise you this is our most technical slide here. We can look at first the prokaryotic cell with the bacterial, long DNA, circular DNA plasmids floating around, versus chromosomal DNA with base pairs that become coiled in a double helix and then supercoiled into all of these different stages, and then become a chromosome, which is then in the membrane-bound nucleus. So the plasmids are really cool in that they can be full little codes for a specific protein or a specific enzyme. And one of the go-to ones that we can think of here like this is insulin. And so in insulin production, we actually take bacteria and introduce them to the plasmid which produces insulin, and then encourage their proliferation. We create the conditions necessary for them to multiply, and in their multiplication and their production of insulin, because of the plasmid that we put into them, we then can extract insulin. And that's kind of a human use of natural processes. But that process of engulfing a piece of DNA, a plasmid, and then creating the protein, if it's useful to the cell, happens with all the bacteria, or with many bacteria in the soil all the time. And so you can have this really interesting evolution of bacteria picking up new plasmids and expressing new genes. So that was sort of a different type of reproduction, evolution sort of thing. A more traditional type of evolution that we're accustomed to learning about, I would think, is sexual reproduction, and like flowers with your anther and your filaments and your stigma and all this jazz. So here we have the floral bracts again, sort of this cool developed leaf with the central part, which is actually the sexually reproductive part of the flower. And so then here we can see some pollination. Those are bees around the white anthurium flower, pollinating it, so that eventually you get these swelling of seeds that can eventually become viable, but they're difficult to plant. So with anthuriums, you often plant them, you often propagate them via cloning and just take a bud that has come off the plant. But I'm in the process of trying to figure out if I can get these to grow from seeds like this. So with that, I want to say thank you for your time and open the floor for any questions. Um, yeah, and so if you want, we can, I also have a fun little exercise prepared that you can take part in a small group discussion or writing if you would like. Cool, so this is the little exercise that I mentioned. It's called micro minutes. And okay, great. I've got my little picture out of the way there. And so as single-celled organisms, microbes don't really have gender or sex characteristics. They literally pick up DNA floating in the soil solution, and they can add it to their code when they reproduce via mitosis. The code then would be in both of the offspring cells. You know, and of course that happens once they've collected sufficient nutrients and energy from carbon or whatever energy source they use in the soil. So in the process of obtaining the nutrients they need, they interact with the soil ecosystem, solid particles that we talked about, either organic matter or mineral, air, water, plant roots, fungi, and other organisms, including nematodes and earthworms. So what I would like for you to do now is to take the next few minutes to write or discuss what a microbe's existence might be like. Feel free to indulge in some personification or go more technical and scientific, or even treat it just like writing the minutes of today's class. So regardless of what you choose, I hope you have some fun enjoying the next few micro minutes. And then the last few things here I have are a couple introductory slides to some of the forest friends, which I think is just kind of fun to help you kind of get an idea of being in the forest and, you know, things. Yeah. So this is my forest friend, Pink Toes, the spider. So Pink Toes, the spider, happened upon me when I was trying to put on my boots. Hence, my socks are there on the side. And I'm not gonna lie, Pink Toes is a semi-poisonous tarantula. And I was not interested in interacting with Pink Toes. You can see my boot there. I ran out of dodge from the ledge where I was trying to sit down. Yeah. So then we also have Little Snake and Big Beetle. I just used the little and big because it was funny because the beetle is really about the size of my thumb, and it was thicker or as thick as the snake was. Yeah, so the snake I came across when I was there moving some wood to create the growth line, the kind of beds for the anthuriums. And, you know, again, I got out of Dodge. I couldn't identify the snake. And either way, I said, you know, there's other places for me to be. There's other chores and tasks for me to do on the farm. I don't need to tangle up with that snake. And, you know, that sort of is a part of my philosophy of being up in the forest as well. That, you know, you got to find a way to share the space. I know we have employees who do make it a point to get rid of venomous snakes. And you know, that is what it is. I kind of respect that position, but I do not hold that same position of wanting to kill snakes because I think they could be poisonous. Um, you know, Sangre Grande Hospital has one of the best venom units and also just has great general health care. Um, so we're safe and we're close enough to Sangre Grande that, you know, should anything really bad happen, we'll be okay. We'll get there in time. Here's a little thing about alternative forest products. So, you know, we can grow plants, we can sell flowers, but one of the things we can also do is kind of find a way to make the most out of what we happen across in the forest. So here are a couple videos of me we were cutting some trees because we needed to clear some area to do some farming. And so this is a guacano tree stump. And I just looked at the stump and I was like, whoa, the bark started peeling in little pieces, which I'll hold up there on the left-hand side. And I thought to myself, what could we do with this? This is such an interesting thing. So I was kind of inspired by a class I once took about Australian Aboriginal art, where they actually use bark parchment to create art, and also for even like really cool legal documents. And there's one treaty that's signed between the Australian government and like one of the Aboriginal groups about their land tenure and stuff like that. So I was inspired to try and see what I could make happen. And so this is how it started. This is how it's going, and this is the inspiration for my art, bark art. So with that, I want to say thank you again for your time, and hopefully we get to see you sometime soon at Grosvenor. All the best.